Thank you very much for coming, and thank you so much for inviting me. It's a great town, a great university, and a great company, and lots of friends around here, so it's great. So this is the title of my talk. That picture is from Davis. I love this. I bike around here, so I thought it was a great picture. But I will try to give you a little account of what we are doing for the last 5 years. And I wish I could have more explanations. We have a lot of results, great results, I think. And we're understanding how things happen. But only in the last year, we start seeing a little bit the light on the mechanisms. That data is not included, but that will be nice if we can discuss it. So ask me questions after the talk. So you can read this, so I will just tell you. Essentially, this came, I remember even when, it was 2005, and we were in Adelaide, Australia, and it was hot. And we, in a meeting, what we do in the afternoon, we go for beer, and we are sitting outside and talking. And we were talking, as we are talking with my colleagues, we see this plant planted outside the little coffee shop, and it's dropping leaves. And I remember I tell my colleagues, this is just a stupid strategy. Because we know what the plant is doing. This plant learned during evolution, hundreds of millions of years, to avoid stress, drought in particular. In order to keep water, they drop leaves, senesce very fast, and reduce the area of evapotranspiration. That's great. During evolution, that has been shown to be a great, successful strategy. Notice you are crops. We invented crops. We did it. 10,000 years, 12,000 years. So we don't want a crop to drop leaves and put very few seeds. We need yield. And I say, that's stupid. Really, we should try to inhibit maybe that stress-induced senescence. And that was essentially the basic story. Our hypothesis is, if we could inhibit stress-induced senescence, we believe then that we could improve the source-sink relationships needed to survive stress. Because in that leaf that is dropping prematurely, you have potassium, you have nitrogen, you have phosphate, you have even water there. They're not totally white or yellow when they drop. So the idea was, okay, let's see how we do that. And the first thing that should come to the mind of any physiologist is cytokinin. The relationship between cytokinin and senescence has been known for years and years and years. So I said, well, if we could express, say, IPT, which is the bottleneck of cytokinin synthesis in plants, we could have maybe stress-tolerant crops. That's not new. That has been done for so many years and in so many things. Let's go like that, will be easier. In potato, and tobacco, and maize. And it didn't work very well. The reason is that people that try this, they try constitutive promoters or senescence-activated promoters. When the leaf is just senescing, then the promoter is activated, like in the case of SAG12, to produce cytokinin, but then it's late. Why it's late? Because plants will survive the stress, but phenology was totally changed. Flowering time was totally changed. Signals for flowering were not coming. And as you know, if you do agriculture, one of the things you cannot change is the length of your crop. If you are a farmer, you are depending on rotation. You cannot go 2 extra months or 3 extra months on your crop just to get some yield. That will be terrible. So the other problem is that if you go too late, the senescence leaf now stops senescing. That's great. Now the bottom leaf is how I don't get blind. Yeah. So the bottom leaves are incredibly young now. They're reverting the process, and we get what is called the source sink inversion. These leaves do not degrade chlorophyll, which is very important for nitrogen homeostasis to the sinks. They do not pass potassium, they do not pass phosphate, and then all the flowering process is inhibited, and also seed setting, filling, etc., etc. So the problem is, maybe the idea is good, but what we need is a promoter that can be activated before senescence. And we start looking for promoters like that in the literature. And I was lucky that in that table in Adelaide, Shimon Gebstein from the Technion Institute was sitting there. He was one of my teachers, and says, and I say, that is a main problem. And Shimon says, hey, you know what? I have a promoter for you. And he offered me his promoter, and he published, we just published a paper doing all this. We can talk about this later, but this is called PSARC, and SARC is senescence activated receptor kinase. He does not have, neither do I, a foggiest idea of what this gene does. We don't care. The most important thing is what Shimon showed, that he showed that the expression of this promoter, and this is in peas, that the expression of this promoter is self-regulated. Whether it's attached or detached, you can see that it's self-regulated, goes up and comes down. And it does it before senescence. Very important. So what we did is just transform it, just put, SARC and the IPT and the control of SARC. We use Picambia, we transform tobacco, and we publish this. But essentially, this experiment was done. Those are the wild-type plants. These are transgenic plants. We grow them, and we stop watering for 2 weeks, close the greenhouse, and left. We don't want whiteflies there. We came back, we water for a week, and this is what happened. I think that the differences are quite obvious. Well, in the greenhouse, everything works anyway, but the differences are very, very obvious. We went to the field, and I hope you guys know it's very difficult to do a drought experiment in the field. It's just terrible. And I promised myself I'm not going to do it using limited irrigation anymore. The way we did this in Imperial Valley in 2009. I don't know if you know the Imperial Valley. It's in Southern California, near Mexico. The snakes do not come at noon. They come up at 9 a.m. because at 9 a.m. already is 100 and something Fahrenheit. So it's very hot. When I said that we grow tobacco there, people were laughing. Nobody grows tobacco there. But nonetheless, the way we did the experiment was we calculated the optimal watering that we need, and then we did half of that amount, a third of that amount, and a quarter of that amount, just in the field. And then you can see clearly that when you compare just the wild-type plants between 100% and 50%, the plants do not realize, this is not significant, they do not realize there is water stress. The problem there is heat. It's too hot. Nonetheless, when you go below, you can see that at 25% of the optimal watering, you get a 50% reduction of yield. Here we are talking about foliar mass. In tobacco, you cannot do foliar mass and seeds in the same plant. You have to do it separate. And you can see what happened on the two transgenic lines. Work quite well. The effect of drought was, the water deficit was less effective in reducing yield than the transgenic. Tobacco. So we decided to do it in rice, and for rice, we try to do physiology. When you go in crop plants, in particular rice, or any of the monocots, you know that you want to see stress in the reproductive stage. This is a critical stage where, and I think that John already mentioned that, this is a critical stage where you should measure this. And our strategy is very simple. We are doing this in the greenhouse, so we water. We stop watering, say, at pre-antithesis, and we rewater only when we see stress symptoms in the transgenic plants. The wild-type plants are already suffering. We don't care about them. When suddenly you see these typical symptoms in the transgenic plant, we rewater. We did the same thing at post-antithesis. And just to show you the expression of IPT in rice, you can see very nicely how it's really expressing during the reproductive stage. Another thing which is very important is that the promoter is also stress-induced. We wrote extensively about the importance of using promoters which are induced by the stress. You don't want to get penalties just because. And you can see nicely in our experimental set, we're talking about 2 lines here, and you can see that the 3rd day of stress, the induction goes up and comes down, and you can see nicely flag leaf, 2nd leaf, 3rd leaf, 4th leaf, and even some expression in the roots. This is nice. We are getting a promoter, which is maturation induced, but also stress induced. And you can see what happened to photosynthesis. Those are the plants well watered during stress, wild type and IPT, and rewater. You can see the difference between the transgenic and the wild type, and you can see photosynthesis in the wild type severely inhibited, but not in the transgenic plant or during stress or well water. Regarding yield, well, the difference is obvious. This is pre-antithesis, this is post-antithesis. And you can see those are 3 independent lines. We are getting around 30% loss of yield versus, I don't know how much, 60, 70, 80% in the wild type. We measure gene expression and changes of gene expression. Sorry. This is a wild type, well water, water stress, the transgenic, well water and water stress. And one thing that appears obvious is that there is an intensive crosstalk between the different hormones in the plant. We knew that. It's not our invention, has been published extensively. And you can see that, for example, genes involved in cytokinin, you can see very clearly in the transgenic plants the expression. Red is more expression. Regarding ABA, you can see a lot of differences. And also, this is interesting, when we measure ABA, you can see the increase in the transgenic plant versus, in the wild-type plant versus the increase in the ABA in the transgenic plant. We know about the interaction between cytokinin and ABA, the negative interaction, and obviously it's playing a role there. And we see Brassinosteroids, and something very interesting, because we are finding actually that Brassinosteroids are much more important for seed filling than cytokinins. And we have now generated, you can see the expression of all these genes associated with Brassinosteroid synthesis and regulation. They are going up in the transgenic plants. And we have generated now, oh gosh, we have like 14 of these genes in Brachypodium and in rice growing, and we are collecting seeds and trying to phenotype them. You can see the same with jasmonic and salicylic acid. There are changes. You can ask me, hey, maybe you have a drought-tolerant plant. What happened with the tolerance of that plant to pathogens? And I have slides for you in the question period, if you want, that shows that there is no effect on their tolerance to Xanthomonas. And the same with all the other hormones. You can see a very clear and distinctive change in expression between the wild type during stress and the transgenic plant during stress. What happened to why we grow rice? We grow rice because we want starch. That's what we collect in rice. So what happened to starch? And for example, if you measure sucrose, you can see that there is more sucrose in the flag leaves during stress. This is well water and this is stress. Starch, you can see that although there is a decrease in starch content during stress, we can see that there is, this is the flag leaf, please, and you can see that there are very significant differences between the wild type and the transgenic. And if you go to the grain, well, that's a very important message, because in the grain, we end up with 30% more starch than in the wild type, even at wild type conditions. This is actually, we were surprised of seeing so high, and was a good news. The best thing that can happen when you grow rice is that the grain has more starch. This is in the greenhouse. What happened in the field? In the field we are trying. We have a collaboration with IRRI going on. They are trying with the IRRI varieties. We are trying with the boring varieties in the field, and sometimes it's not so easy to work on stress on rice. Regarding starch, you can see very clearly that there is a decrease in expression in all of the genes associated with the starch degradation pathway. And I'm going to come back to this in a moment. And now, actually. The next slides are very complicated. Each of these slides can take me half an hour. So I'm going to go fast, and you can ask me. Here I'm showing expression, but this, you see there are differences in expression. Let's leave it like that. What we are trying to see is what happened with enzymes. So we're trying to do all of it. We do the genomics, and then we do some metabolomics and proteomics, but you know, you need enzyme activity. You have to go to the, break down the tissue and measure enzymatic activities, because you need that. And for that, you can see the message here is very clearly in the transgenic plant. I think that CSU has to buy a new projector. Yeah? But this is, I don't know if you see it, this is high. One of the things which is very critical is that in the wild type, we have a decrease in sucrose synthesis and an increase in sucrose degradation. This is sucrose synthesis going the other way, and that is very, very clear. It's very high here, and it's low here. And also, there is a big change in cytosolic invertase. As a consequence of that, I'm making it simple just to go fast. As a consequence of that, if let's do the next one, will be even easier. As a consequence of that, what you have is a huge increase in fructose and glucose in the wild type. This is the flag leaf. In the wild type, during stress, you can see that fructose and glucose are not going up in the transgenic plants, and neither sucrose. The difference is very, very obvious, but you can see the increase in starch. What we are seeing here essentially is that the wild type plant is trying to osmoregulate. One sucrose, one molecule, two molecules, fructose and glucose. There's a need of osmoregulation. They are using their sucrose as much as they can because they have to osmoregulate. And while doing that, well, there are other problems we are going to see in a moment. That's not enough. And of course, your starch goes down. You have to use it. You have to remember starch does not go up and down. It's sucrose that goes up and down. And you can see the relative increase of starch in the transgenic plant. clearly, I'm only repeating these graphs, sucrose phosphate synthase and sucrose synthase doing degradation. Now, this is a slide which we can be all day here. So remember this picture. Making this slide took us like a week. This is incredible. So much data is there. Let's try to go step by step. And okay, let's do it short. One thing that you can see clearly is that in the wild type, during stress, this is the flag leaf again, this red arrow points to amino acids. The leaf is full of amino acids, the wild type, the one that is not going to survive, the one which is suffering. Amino acids are all over the place. But we will see what happened to protein synthesis later. The other thing which is very, very clear is the great increase in nitrate in the transgenic plant versus the wild type. So what you have here is, on one hand, full of amino acids in the wild type under stress, low nitrate. In the transgenic, no amino acids. Well, there are amino acids, not so much, and a lot of nitrate. And that is important because we are going to talk now about that relation between carbon and nitrogen assimilation, which essentially is what makes a plant grow. If you don't have protein synthesis, forget it, you are not going to grow. And you have to regulate that carbon and nitrogen synthesis during stress. So, more expression, and listen, the take-home message is this right here. You can see that all of the genes encoding enzymes in carbon assimilation, TCA, glycolysis are up, or most of them. And you can see also increased expression of enzymes related to nitrogen assimilation. And also, for example, you can see number 5, phosphate phosphoenol pyruvate translocator, for example. These are very important to shunt energy to the chloroplast. This chloroplast is healthy, is doing well. And now we can see some of the amino acid transferases. There is some increase, and this is very important because we know we are assimilating all of the amino acids into proteins. And please, these 2-oxoglutarate, alpha-ketoglutarate in the past, and GDH are critical steps to link this side of the diagram, nitrogen assimilation, and this side of the diagram, carbon assimilation. And what we are seeing, and you can see it very clearly here, that deamination in the wild type is going very high. Why? Because they have a nitrogen deficit. So what they are doing, they are consuming all the glutamate, shunting into oxoglutarate, hoping that this is working, but it's not, and producing ammonia to try to feed assimilation. But they cannot. They are doing this, but it's not working. With the transgenic plant, it's no problem, but you can see the activity of nitrate reductase is quite high, nitrate is quite high. These guys are assimilating with no problem whatsoever. My students love this web diagram. I don't understand it very well. But essentially, what you can see here is when you compare wild type with transgenics, you can see that we have a lot of starch, a lot of sucrose, nitrate, a lot. And this is very significant. We have a lot of glucose 6-phosphate. There is a lot of energy going on. And we have to remember that the pentose pathway is working really well in the transgenic plant because you need NADH, you need NADPH, you need reducing power to assimilate. And they are providing the energy. This was, and it's becoming more and more interesting, but during the discussion I will tell you about this, the protease activity, we find a huge increase in protease activity in the wild-type plants during stress. With the transgenic plants, you can see that essentially it's not very different. And also an increase in protein carbonylation that precedes protein degradation. It's very, here we are measuring carbonylation. And, oh, more of this. Listen, look at this. It's the most important. Lots of protein synthesis, genes associated with protein synthesis in the transgenic plant. I'm going to come back at protein synthesis and what that means for stress. So, listen, third leaf and flag leaf, we're trying to understand source sink here. And essentially, here is very easy. What you are seeing, we are comparing transgenic versus wild type, and you can see essentially that the wild type is senescing, period. It's senescing because you can see that SPS is going down, is up in the transgenic. Sucrose synthesis is going up in the transgenic, obviously down in the wild type. There is less activity of GOGAT. They're assimilating less. They're producing not producing sucrose anymore. They're old. During stress, what you can see clearly, and I just make this in order to make it simple, the discussion, clearly in the transgenic plant what you have is that the cytokinin-induced sucrose synthesis. There's much more primary nitrogen assimilation because we know about nitrate reductase. You saw the amount of nitrate in the plant. We have less reassimilation, less production, what I call deamination. And I'm sorry if there's any biochemist here. I always confuse deamination with reassimilation. My students don't like that. But for me, this essentially, you use all of your amino acid to produce ammonia when you are running out of nitrogen. And the transgenic plants don't have to do that. They don't need it. They are assimilating very well, thank you. And, of course, you see reduced sucrose degradation. In the third leaf, you still see a sucrose synthesis because you have to pass sucrose. You see less degradation, and of course, you see more assimilation. Essentially, this is what I believe is how a proper sourcing relationship should work under stress. Cytokinin is doing it, and we don't have the foggiest idea why. We have some hints, and I will talk about that later. Okay, I have time, no? Good. What we decided is to see if this strategy will be also good for salt stress, with the caveat, we know that in rice, the most sensitive stage of rice to salt is the seedling stage. Here, we just start watering with salt at the budding stage. The plant is already grown. And I think that my students went a little bit medieval with the poor plant because they went up to 120 millimolar. I have to say that this plant will look like this plant a month later. We are delaying. We are not avoiding the stress. We are delaying it. But we are delaying it at least 3 to 4 weeks. This will look exactly like this. So you can see the yield is significant. This is the greenhouse. And this was very interesting for us. We measure the ions because we do transport. And potassium is quite high in the transgenic plants. And to our surprise is, look at sodium in the flag leaf. In the wild type room, very, very high. But look at the transgenic plant. So what we are doing now, we did, gosh, we prepared, I don't know, 144 primers for real-time PCR. And we did qPCR of all the transporters we know in the roads. And we are having really fun. We don't understand, but the differences are incredible. So we're trying to, I cannot show you the data yet, but there is very much differences, not only with HKT1, with the other ones, and in the root and in the flag leaf. If you compare the transcriptomes, obviously, look, the wild type, this is the wild type under stress, the differences are obvious. Look at the two transgenics. They look more or less with some differences. When we go and we try to see how many genes have changed, well, during drought, we detected in the wild type 1,072 genes changing. In salinity, almost 3,000. How many are common? I guess, oh, there will be at least 50%. Nah, look at that. Only 96 genes are common on this. We have the data. We're trying to analyze it. You are interested. We can share it with you. It's so much. Alone, we cannot do it anyway. So, forget this. So what we try, when you go to the field, essentially, you don't have only drought. You have drought, you have heat, and then if the soil is drying, you have a salinity component. So what we decided is we start watering these plants from the fourth leaf stage with 25 millimolar sodium, all the way. And you can see the differences here, and you can see the differences in yield. This is in the greenhouse again, and you can see salt, drought, and salt and drought. So there is quite a significant effect for the combination. I will tell you what we are doing now with salt, drought, and heat at the end. So, very important element. Remember the amino acids? I decided just to measure protein. And look what happened with the protein content in the transgenic plants. It's just high. And this is critical. That's the reason that amino acids are low. Maybe it's not too important for rice, could be very important for wheat, but imagine alfalfa. So what we are doing now is doing a lot of alfalfa, because if this holds with alfalfa, wow, this is important. Remember, alfalfa is one of the main protein sources that we use to feed animals and so on. In wheat, we are getting some differences. You can see the difference in salt are incredible. Again, this is in the greenhouse, and we start salinity at the reproductive stage. At the reproductive stage, of course, we go gradually. We go 40, 80, 100. But every 3 days we change. But the difference is very big, and it's it. We are trying to do now field trials for wheat. We did it last, not we, there's a company, the University of California, of course, licensed the technology, patented the technology. In California, it's a university. They licensed the technology to a company, and this company is trying it. They got some results in wheat which are not bad, are in the teens of improvement, but they're doing it in Idaho, I think. They're going to try this here again. The only limitation that we have that we do not have too many transgenic lines, because we did bombardment. There's no agro in wheat yet. So let's go to the field. And I got so tired of field trials with no budgets. It's so difficult for us. I don't know how many of you guys try to do field trials with very limited budget. It's impossible. You try once, and in California we had rain last July, if you remember, and the year before in June. So everything went kaput. So what I did is I called Hong Shang from Texas Tech University. He's a great friend of mine. I say, Hong, you are in Texas. You guys are dry, per definition. Let's transform peanuts, and let's do the experiment in the field in Texas. I don't have to worry about watering or watering. Whatever happened, happened. And this is what we did. We just published this. You can see one thing immediately is we are measuring here the number of pods with 3 or 4 seeds. And if you believe whatever I show you and I said about nitrogen, you will expect more pods and more seeds, because that's where the nitrogen goes, in peanuts at least. And well, then look at this. the differences are very clear. We get bigger pods containing better seeds, more seeds. Quality of the peanuts is great. It's the same quality. We did the analysis that USDA did the analysis for that. So we went to the field, and you can see this was in 2009, but we have a problem with the irrigated one, so I cannot show the irrigated one, but you can see the comparison in 2010 between irrigated and the dry land, and you can see that the yield penalty in some of these lines is only of 30%, totally dry, no water, whatever happened, and versus 60-something% in the wild type. This is a good result. Growers there were very, very, very happy. Of course, in 2001, there was only 12 millimeter rain during the season. So really, nothing grew. But we went to cotton, and you can see nicely the difference in photosynthesis activities, stomach, blah, 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 between the 1, 2, 3, 4 lines of transgenic, null, and the wild type. And then we measured cotton ball and other parameters, and you can see the difference very clearly. And this is in a field that was low irrigated, a USDA field in Lubbock. But then we did it also in dry land. The yield, as you can see, is lower. No, the yield comes in the next slide. The yield is lower, yet the differences are very, very significant. Mind you, in 2011, the wild type, the transgenic cotton was 14 centimeters, and the wild type was 6. Were great, but you cannot measure when there's no water at all. There was no rain at all, so we can everything. Other things we are doing, well, we are, in the greenhouse, we are doing corn, walnut, leptidemia. I'm going to explain this in a moment, turfgrass, brachypodium, in the field, canola, wheat, alfalfa, and all this. Most of these are done now by a company, not by us. We just let the baby get out of the house. But you can see something nice. Working at Davis, you have to work in pomological crops, and this is walnuts, and you can see nice the null and the transgenic after 10 days of no water. This is just a wild type well water. Of course, 20 years from now, we are going to know about the seeds. We have to do it. But this is also very important. This is a bush that can go up to 3.5 meters tall. It's called leptidemia, leptidemia pilotechnia, grows in the Arab Peninsula. I chose the seeds there, and the idea is, can we use these as a tool for afforestation? Afforestation means we want to change the dunes. And the reason is, this is very drought tolerant. The only problem is, if you want to grow hundreds of thousands of these to plant, and we have a method to do that, you have to put them in a lat house for a year or two before you plant them in the field. When they are in the lat house, they are very drought tolerant. And you can see nicely the phenotypical difference we are getting. Seeds, we don't expect seeds from this. We don't produce seeds so easily. So, just to finish, this is the strategy that we are trying to apply now, mainly in rice and brachypodium. We are trying to now combine PSAC. We try always to keep it away from the 35S with NHX1, a gene that we show increases the sodium accumulation in the vacuum, providing some salt tolerance, and another gene that we cloned from Candida is an itch shock factor that promotes great stability by regulating itch shock protein expression. So those are the concepts that we are trying right now. And this is the people that they work. You can read all the collaborators. These are the students. We already do the job, Rosa on the job, Armando on the job, Maria is still there, John Kuhn just arrived, is great, and some of the other people, the collaborators I have, as you can see, from everywhere. Without them, it would be impossible to do the work. Thank you very much.